Hello everyone, welcome to the new session of Mendel's Laws of Inheritance. So this is the third part and we are going to study about the third law of Mendel, that is the law of independent assortment. So by definition, the inheritance of more than one pair of characters. So we in the earlier the two law we consider one pair of character, right? One pair of character. Either we were considering the height, that is denoted by capital T and small t. For example, this is the height of plant. We can also consider the color of flower. So we have taken the example of purple, that is capital P, and white flower denoted by small p, right? But here for the independent law of assortment, right? Law of independent assortment, we are going to take more than one pair of characters. Okay, so two pair or more is studied simultaneously. The factor or gene for each pair of characters is sort out independently, means they mix independently and make different combinations, right, with other pairs. So we are going to study about this. Mendel formulated this law from the result of dihybrid cross. So till now we have considered about the monohybrid cross. So capital T, small t. This is the mono. If we are making the cross between them, then it is called as a monohybrid cross. This is for one character, right? If it is for two character, right, simultaneously, then it will be called as dihybrid cross, right? So let us see with the example. Now the explanation of this: the cross was made between the plants having yellow and round cotyledons and plant having green and wrinkled cotyledons, right? So the F1 hybrid all had yellow and round seeds. So in this case, they all have yellow and round seeds. Okay. Explanation: when these F1 plants were self-fertilized, they produce 4 types of plants in the following proportion. So they are having yellow and round, yellow and wrinkled, green and round, green and wrinkled. So we are having 4 different types of combination, and that is in the ratio of 9:3:3:1, total 16, right? So this was the law of independent assortment by one of the example. Okay, then explanation of this: the above result indicated that yellow and green seeds appear in the ratio of 9+3 and 3+1. So what will be the correct number? 9+3, that will be 12. The ratio will come in this form, 3+1, that will be 4. So when you simplify it, you will get 3:1. Okay. So similarly, the round and wrinkled seeds appear in the ratio of 9+3:3+1. So then you will get again 12:4, and when you simplify it, you will get 3:1. This indicates that each of the two pairs of the alternative characters. Okay, so the above experiment, yellow and round characters are the dominant over the green and wrinkled. So round and yellow, the color and the shape, round and yellow. So that is denoted by RR, okay, round, and yellow, that will be Y and Y. Y and Y. And the green and wrinkled, so green cotyledons will be represented as small y, and wrinkled, that will be small r. So that will be the green and wrinkled. Okay, green and wrinkled. And this will be round and yellow, right? So the gene for the yellow color here, the cotyledon represented by Y. Green color is of the cotyledons by small y, and the capital one, that is the yellow color. The round character of the cotyledons represented by the capital R. The wrinkle character of the cotyledon was represented by small r. Okay. Therefore, the plants with yellow and round cotyledons will have their genotype capital Y, capital R, and those with the green and wrinkle of the cotyledons will have genotype small y, and small r, right? So these plants will produce the gametes. So let us take this in this way: capital Y, capital R, small y, small r. Okay, so what type of gamete they produce? They produces capital Y, capital R, capital Y, capital R. Okay, and this will produce small y, small r, small y, small r. So generally this one will have capital Y, capital R, and this will have small y, small r type of gametes. So when they fertilize these gametes, when they undergo fertilization, they will produce capital Y, capital R, small r, right? So like this. When these plants are cross-pollinated, so for in the F1 generation, we will have capital Y, capital R, small r genes, right? So we will have capital Y, capital R, and small y, small r. So they will produce capital Y and capital R type of gametes, and in this case we will get small y, small r type of gametes. So when these gametes fuses, they produces capital Y, capital R, small r type of plant. So that is the F1 generation. This is the F1 generation, and this is the hybrid plant. Okay, so when these produce gametes, all four genes have full freedom to assort independently. So this one, capital Y, small y, capital R, small r, this will have four different types of combination. And what will that? Capital Y, capital R, capital Y, small r, small y, capital R, and small y, small r. So we will have four different types of combination. And why this four different types of combination has been formed as a gametes? Because they have a full freedom to assort independently. They have full freedom to have a different combination independently, right? So this is the these are the possibilities of the four combinations in both male and female gametes. So male will have these type of gametes as well as female. So we will have this checkerboard or the Punnett square, and we will have male plant and female. So they will produce gametes like capital R, capital RY, okay, capital R, small y, small r, capital Y, and small r, capital Y. In the same way here, capital R, small y, small r, capital Y, and small r, small y. So we will have these different types of gametes, and they have full freedom to unite with the different types of gametes in this way. Okay, so we will have capital R, capital R, capital Y here, capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Similarly, here we will have, okay, capital R, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Now here we will have capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small y, small r, capital Y, small y, small r, small y, small y. So we will have these 16 combinations, right? These 16 combinations. Okay. So this shows the excellent example of the independent assortment. These gametes can unite at a random, producing it all 16 different combinations of the genes, but presenting 4 phenotype. But their phenotype, how they appear, that will be just 4 types, and that will be in the ratio of 9:3:3:1. Okay, so let us see that. Here we are having these combinations as we have seen earlier, right? So we can have this round yellow, because for round, either both R or single R has to be present. Capital one. Both R should be capital or single R should be capital. So that will be round. Okay, so these are all round. Okay, now for yellow, again, that has to be both Y capital or one single Y capital. So here both the Y are capital, so yellow. Single Y, yellow. Single Y, yellow. Both the Y, yellow. Single Y, yellow. Single Y, yellow. Both the Y, yellow. Single Y, yellow. Now let us see this one. Round. For round, both the R should be R capital or single R, right? So here round. Okay, but in this case, this is also round and yellow because one R capital and one Y capital. But in these case, both the Y are small, right? Both the Y are small. So they will be green. Round and green. Okay. Now here comes. Both the R smaller, but one of the Y is capital, or in this case, both the Y is capital. So here three individual will be wrinkled and yellow. And the one that is having small r and small y, that will show the green and wrinkled seed. Okay, so this, if we count the round and yellow, so we will have 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9 out of 16 is round and yellow. Let us see this green one. Round and green. Round and green. So 1, 2 and 3. So that will be 3 by 16. And if we see this wrinkled green, wrinkled yellow and wrinkled green. So what we'll find? We'll find that this wrinkled yellow 1, 2 and 3. So 3 by 16, and this is only 1, that will be 1 by 16. So what will be the ratio? The ratio will be 9:3:3:1. So total 16. So out of 16, 9 are round and yellow, 3 are round and green, 3 are wrinkled yellow, and 1 is wrinkled green. Okay. So, now test cross. The test cross is an experiment first employed by the Gregor Mendel in his studies of the genetics of traits in the pea plant. So, Gregor Mendel was using the pea plant for his different types of hybridization by taking the pea plant, right? So, he also done a test cross. Now, what does the test cross say? The test cross says that the plant of F1 generation, is crossed with recessive parent. So that is called as test cross. And when the F1 plant or the F1 generation plant is crossed with any of the parent plant, whether it is a dominant or recessive, then it is called as back cross. That is called as back cross. But specifically when we are taking F1 plant and the recessive parent, then that will be called as test cross. So a test cross is a breeding method that entails pairing an individual with an unknown genotype with another individual that is known to be homozygous recessive. So here this homozygous recessive is the recessive parent, right? And this is the unknown genotype. So if you get this 1:1:1:1 ratio, then that will be the heterozygous. That is the heterozygous. So test cross can be used to determine whether the dominant phenotype is homozygous or heterozygous. If the unknown parent is homozygous dominant, all offspring will express the dominant phenotype. And if the unknown parent is a heterozygous one, half of the offspring should be the dominant and half of it will be recessive, right? So here, if they are all dominant, then you will get homozygous dominant. All the dominant phenotype you get, then it will be the homozygous dominant. So the law of independent assortment failed to have a universal applicability, right? Cytological studies have revealed that only those allelomorph assort independently during the meiosis, which are located in the different homologous pairs of the chromosome. Different homologous pairs of the chromosome means their locus on the chromosome should be different. Okay. But if the allelomorphs for the different character are present in the same homologous pair of the chromosome, these are passed to the same gamete. So the law of independent assortment does not apply to such cases. So this is all about the Mendel law of inheritance. So that is we discussed today, that is the law of independent assortment. Okay, the law of dominant and the law of segregation we have already discussed. So today we have completed the Mendel's law of inheritance. Thank you very much.